Welcome to Data in Biotech, a podcast from CorrDyn where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. This week, we're excited to be joined by Harry Rickerby, co-founder of Briefly Bio, a company that helps scientists capture, share, and collaborate on their experimental methods, kind of like the GitHub for experimental science. During this conversation, Harry shares the origins of the Briefly Bio platform, stemming from his frustration to get even the most basic assays working reliably in the lab, and how this resulted in developing a solution to improve experiment documentation and reproducibility in science. He also shares how large language models are used to facilitate experiment protocol structuring in the platform, his perspective on the limitation of LLMs in highly specialized contexts like scientific fields, and how metadata is generated within the experimental design process while interacting with an LLM. Here we go.
Harry Rickerby, welcome to the Data in Biotech podcast.
Hey, great to be here. Thanks, Ross.
Awesome. Just to kick us off, would you mind just giving a brief background on what led you here?
Sure. I come from a biology background originally, studied at Imperial. I got really excited about synthetic biology, about this idea that you could engineer biology to create meaningfully useful things for humans. I met someone called James Field during my undergrad there and did a project together. And then I joined his company called Lab Genius back in 2014 as first employee. We were focused on using data and machine learning to help with protein engineering, drug discovery. And my role evolved as I was there. I started out in the lab and then moved into more leadership roles. The focus there ultimately for me was looking after, what workflows are we going to bring into the company and the lab side, and then how that kind of interfaced with technology. So what software we would build, what automation we would use, how we'd use data science. I founded Briefly with a couple of other folks last year, and I guess we'll talk a bit more about what that is as we go through. But yeah, that's how I got here.
Yeah, awesome. Since you mentioned it, why don't we just jump right in? Can you just provide an overview of the platform that you're building?
Yeah, sure. So I guess the problem that we saw inside the companies that we worked at, and then also more broadly in the field in general, is this inconsistent documentation of experiments. And that kind of causes problems all over the place. If you're a scientist wanting to run an experiment, you don't know exactly how somebody else ran it, so you have problems with reproducibility. If you're a data scientist wanting to analyze a data set, you need to know exactly how that data was generated in order to be able to analyze it properly. All of these kinds of things. And so the idea behind the company is, how do we solve this problem? How can we get to a place where when you look at an experiment, when you read the documentation behind an experiment, you know exactly how it was run? And so that's ultimately what the problem we're trying to solve. And the way we're doing that is basically building a platform where scientists go to create and design their experiments. And we do take a lot of the grunt work out of that process and do a lot of the structuring behind how to make that information consistent, how to fill in all of the gaps that exist in a lot of the documentation that's out there.
It sounds like a really interesting platform. Did the genesis of Briefly come from some of the problems that you experienced while you were at Lab Genius? And, what kind of things did you notice that you allowed this idea to come into your head as the next thing you wanted to work on?
I think one of the key things that I faced, because I was, early in the company, we were starting from scratch, trying to build this really ambitious platform for protein engineering. And so we had to set up everything from scratch. We built our own lab, and we were getting all of these workflows off the ground from scratch. And I think a lot of the time, what frustrated me was how difficult it was to get even basic assays, basic workflows working reliably in the lab. Even things that you would think, I'm just pulling this in, it's pretty standard. There's plenty of papers published on it. I know people who've done it before. You get it into your own lab, and then you try and run it, and then it doesn't work, and you get into this endless cycle of troubleshooting and trying to figure out how things worked. And your timelines just lengthen and lengthen, especially for someone like me. I was super inexperienced, super naive. I think people who've done this before would know to build in a lot more kind of wiggle room for those kinds of issues. So that was my frustration, and basically just not knowing really how these experiments, how these processes were really being run. You read a paper and there's just not enough information there to get the answer. Often you'd have to actually find someone who's run it before. Even then, you still have problems. And then I guess from the data science side, Katya, my co-founder, was coming at it from this position of, okay, she was purely computational at my last company, and she was getting a lot of data coming in from the lab. you run your analysis and then you see something in the data that looks weird, and like figuring out why things look weird. They looked normal last week. Why things look different now? You have to go down this kind of rabbit hole of figuring out exactly what changed in the experimental process. Was it something in the experimental process? Was it something else? So figuring out all of those answers to those questions, I think, is a lot of the sort of what spurred on founding this company, those frustrations.
Yeah, that makes a lot of sense. So can you just give an overview of how the platform works from the perspective of a scientist who's running a given experiment?
Yeah. The scientist really, we're focusing on that experimental design phase, thinking about what exactly is it that I have to do as a scientist. I'm thinking about my experiment, thinking about my conditions, I'm thinking about my methods. What exactly do I have to do when I get into the lab? What are the steps I'm going to have to follow? What are the things I'm going to have to be tracking? And we're focused on how do we take that sort of phase of a scientist's workflow and make it as streamlined as possible. So often what happens now is a scientist will think about their experimental design, jot a few things down, think about the key conditions, and then run the experiment. And then later on, they might go back and write up the experiment and think about what are all the things I did. Can we get to a place where scientists will design their experiments in this tool, get all of that information into this tool as easily as possible about their experimental design, help them think through all of those steps, help them give them additional information about maybe there are potential pitfalls in their experimental design, potential issues, and then also walk them through, okay, now we're going into the lab. What are the steps I have to take? When am I going to be tracking certain bits of extra information, bits of QC, all of that kind of thing. So it's about that, thinking about my experiment, thinking about the steps, getting all of that stuff into one place, and then running the experiment and following all the steps there.
Yeah, that makes a lot of sense. So why do scientists not document all of those things today?
There's a lot of variability, I would say. Some scientists do document a lot of stuff. Sometimes scientists document less. I think that the truth is that documenting every single step or every single parameter associated with an experiment is the black hole of complexity. It's really hard to know what is the right line to draw, how much detail is relevant. And also an interesting part of it is the more you document, often the less useful that sort of artifact that you create becomes. I don't know if you've ever come across like a Nature Protocols paper. These are like the most incredibly complex, detailed protocols you can imagine. Like every single step is meticulously documented. But actually like getting an overview of a protocol like that is incredibly difficult. And so there's this interesting like trade-off that a lot of scientists are making where they don't really want like a full SOP. They just want, here are the key factors that I need to remember. I've done this experiment before or whatever. So it's like a combination of, it's a huge amount of work to document things. And then even if you do document things fully, you're ending up with something which is less useful at the end of it, especially when you're going into the lab. For us, it's about how do we make that documentation process really fast and easy? How can we shortcut all of the steps to getting all of this information down? And then also, how do we show it to a scientist in a way that kind of gives you an overview? It doesn't drown in the detail, and you can provide yourself with different ways of viewing that information, all of this kind of stuff.
Yeah, interesting. And I imagine that there are benefits to the R&D team as a whole in terms of the visibility into what each other are doing and like how you collaborate on things. Are there features of the platform that are focused on the R&D team experience?
Absolutely. That's ultimately what motivated our development of this platform, right? Is about how do we improve collaboration between scientists, between data scientists, between teams. And so that's all part of it. The real utility of this really good documentation is when you don't have everything in your head anymore, whether that's you in 6 months or whether it's a different person in the team. Part of the aim here is about consistency. It's not just about documenting everything. It's also about how do you make that information as consistent as possible. So if a data scientist wants to say, I want to query this experimental database and I want to see all the experiments were run with these particular parameters, they can search through it. It's not just buried in a blob of text in an ELN entry or in a notebook somewhere. So that's all kind of part of the plan. I think the first thing for us, though, is how do we make a tool which really scientists love to use and they like to document all of that stuff? Because I think until we have something which is really compelling for a scientist to use for themselves, you're then just going to be having to say managers telling their scientists do X, Y, Z. And if the tool isn't useful, if it isn't intuitive, they're probably going to use other things. And then you have the same problem again, like information starts to spread into different places and you don't have everything in one place.
Are you a biotechnology company looking to unlock the potential of your business data? CorrDyn can help. We're an enterprise data specialist that helps companies working in life sciences make smarter strategic decisions. From developing the right data strategy that starts with our data maturity assessment to building and delivering bespoke technical solutions, we are equipped to tackle the most complex data challenges. We have partnered with dozens of high-growth organizations, from manufacturers of custom oligonucleotides to molecular diagnostic companies, to achieve data competency. Whether you need to supplement existing technology teams with specialist expertise or launch a data program that lays the groundwork for future internal hires, you can partner with CorrDyn to unlock the potential of your business data today. Simply visit connect.corrDyn.com/biotech to learn more. Now, back to the show.
It seems like one of the goals of Briefly is to create this great experience for the scientist in constructing this experimental procedure, provides them the flexibility that they need to go in whatever direction they believe the experiment needs to go. But you're also trying to capture all of this metadata, this standard information about how the experiment was run, so that the data team can use that information later on. So I sort of have two questions. One is, why is the data team so focused on getting that consistent metadata? And then the second question is, how do you balance the needs of the scientist in terms of making it a great experience with getting the data team what they need?
I think the question of metadata in biotech is a really big one and a really interesting one. I think there's a big clamor, and I'm seeing it more and hearing it more and more in this space, for we need better quality data. Like in biology, we need better quality data, but also more data, higher throughput, in order for us to understand what's going on in biology, but also if we want to use machine learning, use AI in order to understand, in order for us to unpick some of these really complicated biological relationships. I think the interesting thing about biology and biotech research in general is that the data is not ground truth. It's like the vast majority of it is a proxy. It's some kind of experimental procedure that we have set up, which gives us a window into the biology by making some kind of inferences. And so if you don't have a really good understanding of exactly how your assay works, how your experimental process went, you're really not learning anything about the biology. And I think this is why I guess this metadata question is so important, because if you can change something in your experimental procedure, like it's going to tell you something different about the biology than the last time you ran it. That's quite different from a lot of the other data sets that we use in machine learning. We doing sort of image recognition, understanding the source of that image or the date that it was taken doesn't really change the fact that it's a cat. It doesn't really tell you, it doesn't change what you're learning about that image most of the time. Whereas in biology, it's the whole thing, because your assays are, like I said, they're proxies. It's not the ground truth. And so I think that's why it's so important to the data teams to have this kind of transparency over exactly how experiments are being run. And if you're looking at two different data sets, you want to combine them together, you need to understand what were the differences in the experimental procedure, what effects do those changes have on the data that comes out the other end. And so that's why I think having this structured metadata, it's super important, especially if you want to start to really combine a lot of data sets together, rather than just like creating a single data set for a given experimental question, using it to answer that question, and then moving on to the next thing. If you want to I'm going to take the last 20 data sets and throw them all at this machine learning model and see if we can learn something fundamental about the biology, you need to really understand what are the differences in those data sets, how are they generated. The question of how to make that experience fluid for the biologist, I think, is really the big challenge. The way that it's tackled now is basically biologists and wet lab biologists will have their own kind of notebooks. They'll have their own protocols. These are in free text format, right? They're not structured at all. And then if you have a data team, they might sit down with the biologists and say, okay, which parameters are we going to track? What are we going to put into this big table? And say, okay, these are the things we're going to track over time. I think the thing that we want to say is, let's not make that distinction. All of the process that goes into creating a data set could be important at some point. So can we allow the scientists to create, when they're documenting their experiments in the way they do, creating these protocols, creating these steps for themselves, can we do the work using large language models as the principal kind of technology that we're employing for this particular thing? to allow them to have the flexibility to write things in a way that makes sense to them, but then also to take care of a lot of that structuring side on the back end, so that when the data scientist comes to look at it, it's consistent. You can compare different experiments with one another. Does that make sense?
Yeah, I think it does. I think now would be the right time to ground this in an example. So can you walk me through the step-by-step process for like how the metadata gets generated within the experimental design process while interacting with an LLM?
So the way that we think about this is we like basically went back to the protocol. So like these are the step-by-step instructions that scientists create or companies create for their scientists to follow when they're running a given assay or a given experiment. And we broke that down into some like fundamental concepts that we could structure. So we took the information and structured it. And then the way that a scientist interacts with it right now is there's a number of ways. Like we have that information, like that structured hierarchy is there in our editor. Like it's a tool that you can use and you can build this structured version of your method like from scratch. And you're defining all of the steps and you're defining all of the key parameters associated with it. That's a lot of work. Some of the things that we're doing to take the burden off the scientist in that process of documenting all of these steps is we've built in large language models into the tool from the ground up. We think about it a little bit like some of the sort of co-pilot tools that exist for coders. So you can say, okay, draft me a protocol for X, Y, Z, and it'll give you like a good template to start from where a lot of the information is already structured, a lot of the parameters are defined, and then you can tweak it to the specifics of your own experiment. And on the other side, if you already have something that you're working from, you already have a plain text protocol somewhere or a description of your method, you can also just paste that into the tool and it will do all of the structuring for you. And at the end of it, what you get is this clean, easy to read, easy to understand, step-by-step guide for your experiment that you can then integrate more information into without it drowning in the noise. Obviously, there's lots of other things that we want to integrate into the tool right now. Most principally is your samples. So there are tons and tons of different ways that the companies handle their samples right now, different tools. So we need to integrate with those as well. So when the scientist they have their method, they have their structured method, but then also integrating, this is where the samples live. Here are my different conditions, and look after that as well. So it's all included in this kind of structured version of how your experiment went. Does that make sense?
Yes, it makes sense. It sounds to me like you're rethinking from the ground up the way that the scientist workflow goes, but also trying to leverage the power of these large language models, of generative AI, to bootstrap a protocol together that has all of the metadata elements, and then letting the scientist curate the experimentation protocol that gets generated rather than having to write it all down themselves. So you're getting them to make sure that they fill out all of the elements, but you're also reducing the burden on them to tell you what those elements are. Is that right?
Yeah, exactly. And as we go along, we want to build in more features that are going to help both the scientists in designing that experiment, but also in creating more consistent and more complete documentation. For example, having something which can analyze a protocol and tell you, okay, maybe there are certain parameters that are missing that you should be including here based on what we've seen in the past. Or for the scientist's perspective, spotting potential errors in their protocol, potential pitfalls, or things that look a bit odd that maybe we've seen before in the literature. It's interesting with LLMs, right? They're simultaneously like incredibly smart. They have so much, they've consumed so much knowledge, but they're like the specificity of that knowledge to your own experiment, I think, is often the problem. Like, how do you make sure that they're not just going to give you some generic advice that isn't actually useful for your particular protocol or your particular experiment? So I think we're thinking a lot about that. Like, how can we give you this kind of co-pilot that gives you the right kinds of suggestions and the right kinds of tweaks, and also takes care of the structuring and this completeness problem of all of this metadata that we need to capture.
Another benefit to what you mentioned earlier is improving the reproducibility and the reliability of these biotech experiments, right? So the idea is that this documentation is going to lead scientists to be able to better reproduce experiments that they've done in the past or that other people have done in the past. Do you see that playing into this tool, and how does the tool sort of inform that process?
Absolutely. This is probably the sort of the first reason that we were interested in building Briefly was we thought a lot about reproducibility and I guess the reproducibility crisis broadly in science. And we did a lot of digging into like why this problem exists. What are what are some of the root causes of it, both kind of inside companies and the reliability of experiments inside companies, but also more broadly in academia as well. And I think the thing that we kept coming back to was, so often you don't have all of the information you need in order to actually reproduce an experiment. We want to give that transparency to an individual or to an organization to say, this is exactly how this experiment was run. This is about as much information as we can get. At least this is the intention of the scientist, right? And what they did. There are other things which can be responsible for problems with reproducibility. But I think that until we have some consistency and have completeness in how experiments are documented, it's hard to move on to some of those other issues in reproducibility. A lot of the things that we talk about are, for example, hidden variables. Oh, what was the temperature in the lab that day? What was the lot number of the reagent? Maybe it's gone off. Like all of those kinds of things, I think they're perfectly valid. They're certainly responsible for a lot of issues with reliability and experimentation. But I think a lot of what happens when your experiment fails is you think, what did I do differently? What did I do wrong? And the answer to that question is, or what did I do differently from my colleague? Or what did I do differently from this paper? And the answer to that question is often really hard to figure out. So our kind of first step is, okay, let's get to a place where we have a really good record of that. And then we can investigate some of these other like more hidden kinds of variables and hidden issues that can account for problems with reliability and reproducibility.
Yeah, so ideally, everyone would be using a system like Briefly to generate the operating procedure for the experiment, and then everybody would be on equal footing in terms of all of the things that get tracked about that experiment, so that it would be easier to compare and contrast across experiments.
Right. Yeah. And I think this kind of like ties in with some of the things that we think about a lot, which is things like version control is obviously something that's used in software a huge amount. And version control it's not something that's like really effectively used in biotech. There's some kind of like document control, especially in regulated biotech environments. But if we're talking about being able to say, okay, I want to show me like GitHub, show me all of the differences in the code between this at this time, this release and this previous release, and highlight all of those differences, it's really hard to do right now because the information is incomplete, because the information is inconsistent, it's not standardized. If you could be able to, you could say, okay, show me, this data looks funny. I remember it worked last time. Show me exactly what changed between today and 3 months ago. I think that could help at least answer some of those questions about what could have gone wrong, what could have changed. If things look exactly the same, then you can move on to the funny, the funny unseen variables.
Yeah, so is part of the idea that one scientist can fork the experiment from another scientist and then make some slight changes, but that it would be evident in the experiment itself that it's a fork of this previous experiment. So you could see the experimental lineage of it.
Yeah, definitely. I think the experimental lineage it's super interesting. I think broadly across within organizations and also across like the scientific landscape, it's how like experiments and protocols evolved and changed over years is basically a complete black box. We really just don't know. It's all, a lot of it is like hearsay. There are some papers published every now and again about certain methods, but a lot of things just crop up to solve specific problems and then maybe just hang around forevermore and you never really know why they were there. There's a good example where there's a very common media used to grow bacteria, and every single version of this media includes sodium chloride. And I think they did this big experiment where they were like, do we actually need this sodium chloride? Does it make any difference to how these cells grow? And it doesn't. And this is like pretty much as standard as any kind of biological reagent or protocol gets, like LP media. It's just absolutely everywhere. We all put salt in it religiously, and it does actually not doing anything for most of the use cases. So yeah, having that kind of transparency about, oh, these are where all of these changes came from. Now we can understand why they cropped up. What was the reason for them? And better understand that ultimately, I think would be super powerful. That would be really awesome. I'm super excited to get to building that.
So you ask, why do we use salt? And you get 20 different responses, but the answer is because we always have.
Yeah, pretty much.
Interesting. So what you're doing with LLMs and building your product on the backbone of LLMs is really fascinating, and I think very innovative inside the biotech space in particular. So I want to spend a little bit of time digging in on the way that LLMs are used and how the role it plays in your user interface. So can you just start by providing like how you use LLMs inside the interface together, like where the formal software system ends and where the LLM begins.
Yeah. A lot of the work we've done with LLMs in the user interface right now is modeled on, I mentioned before, a lot of the software tools that are being used for software, like being built for software engineers now, are built on the back of LLMs. Right. And I guess the underlying thesis behind our use of LLMs is experimental design is fundamentally, it's like a creative process. It's about your instincts, your knowledge as a scientist, your understanding of all of the previous data you've generated. All of that stuff feeds into how you're going to run your next experiment. And I think the thing that we focus on most of all is to say, okay, LLMs are a really useful tool, but what is it that they're good at and what is it they're not good at? How do we integrate these into a tool where it doesn't take away that creative element? Like, it doesn't take away your ability as a scientist to design the right experiment. It's not like a prescriptive tool. And so the way that we've built them in is to solve specific needs within the context of what we're building. The first one is, and this is the one we started with, is to say, okay, part of the core of this product is in the structuring of experimental methods. We want to take this kind of mess of how experiments are documented and put them into this like more standardized structured format. LLMs are amazing at that. Like, they understand language very deeply. They can convert between different formats extremely well. And so that's the first thing that we've built in, and we've built it in a couple of ways. The first is like the most kind of basic version of that, is just to say, okay, give me the text that you have, this natural language description of your experiment, paste it into this box, and then the LLM will give you the structured version of it, which is easier for you to, which you can read a little bit more easily, that you can follow a bit more easily. It's nicely structured, nicely laid out. So that's for the scientist, obviously. And obviously it's structured on the back end for when we want to integrate with data teams. Then getting into slightly more interesting use cases where you're leaning a little bit more on the knowledge that the LLM has, like that, the importing feature, like going from free text into briefly format is just leaning on the LLM's like amazing abilities with language. But it also has a lot of knowledge. Like, it understands it read a lot of papers, it knows a lot of protocols, it knows how a lot of science is done. So the other thing we have is like this ability to draft with it. So within the context of the editor, you can say, add a few steps here where I'm washing the cells with PBS. And it'll give you this structured version of the protocol with those steps drafted for you. So you don't have to go through all of the rigmarole of writing it up yourself. And the extent to which that's good is obviously dependent on how many times the LLM has seen that kind of method before. For very basic methods that's seen many times, like a PCR, it can draft you a whole PCR protocol from scratch very reliably. It's like really good at that, and it does it really nicely in our tool. If it's something that's much more specific and much more niche, then obviously it's not going to do as good a job. And you're relying more on this kind of interplay between saying, oh, add a few steps for these particular things here, add a few steps for these particular things here. So that's where we are now in terms of our integration in terms of our use of LLMs with the tool. I think there's tons more that we can do though. They're just like, there's so many interesting use cases, but I think it's about being like really intentional about what it is, like what problem you're solving with the LLM. I've seen a lot of like software products just stick a chatbot into their tool and say, hey, have a chatbot, you can use it to do whatever. And that's really tempting because LLMs are incredibly pluripotent. They can do so many things, they know so much, they can answer so many questions. But I think like you're basically competing with ChatGPT at that point. I think if you're building LLMs into a software product, you need to be like really thoughtful about how it's actually helping your user do the job that they're trying to do. So that's our guiding principle behind our use of LLMs and some of the things we've been thinking about so far.
Yeah, that makes a lot of sense, and it's really insightful. Are you building on the back of one of the APIs, like the OpenAI API?
We're just using the APIs for now with existing foundational models. I think in the longer term, we'll probably want to do some fine tuning and thinking about whether we can add additional like specific kinds of data that are really useful for like train it on specific kinds of data that are particularly relevant for this use case. So far, it's working pretty well with just those foundational models. But yeah, that's something we'll be working on for sure in the future.
Yeah. Are you at all concerned about like the data privacy issues around the sort of protocol data that you're sending up and having the LLMs parse? Or do you feel like these protocols are one degree of separation removed from the important data that biotech companies will tend to care about?
It depends on the company. Obviously, a lot of companies allow their employees to use ChatGPT, but some don't, right? And I think it's going to be an ongoing question for the field generally. How comfortable do we feel with sending this kind of information to OpenAI or to insert foundational model or like insert large language model here? It's going to be something that we think about a lot. I think some companies don't consider their protocols to be at the kind of level of their sort of the data about their molecules, for example, but they're still valuable IP about how they're doing their science. It's going to be something that we're super conscious of and we're thinking about all the time. OpenAI is working on enterprise versions of these models, and obviously a lot of other companies working on it. So it's going to be I think a lot of interesting stuff is going to be happening in that kind of data security with these large language models that we'll have to keep on top of and figure out how to navigate.
For sure. Can you just talk about some of the challenges that you faced in trying to build your user experience around LLMs? Was there anything you didn't know going in about building a UX for that you wish you had known when you started?
It's like a constant learning experience, I would say. The thing that we're learning most of all with this stuff is really identifying what are the specific use cases where the LLM is going to actually help the user. And we have a test version of the product right now where you can just ask the LLM to do things for you in quite a general sense. And I think generally it's tough to know when you're faced with that kind of open, expansive possibility to really know what should I do with this thing? What is it actually going to be useful for? And so I think doing a lot of prototyping, right, understanding what are where does it really excel? And then building features around those particular niches is, I think, the thing that that we're going to continue to do more of. Otherwise, I think you're not really offering a huge benefit over just using a chatbot somewhere else. It has to really be integrated very deeply with the tool that you're building. That's how I'm thinking about it right now.
Yeah. So I imagine as you're experimenting with the tool, you're finding the limits of where the LLM's knowledge ends. Are there any unique challenges that you've discovered with the highly specialized language that you biologists use and the more generalized knowledge that the LLMs have?
I'd say that they're pretty excellent when it comes to the specialized language of biology. They really have an amazing grasp of language. And I think it's because they have this context around the language that so many other tools in the past have lacked. They will generally have a very good understanding of, there are so many acronyms in biology, right? There are so many acronyms. And for me, I always struggled personally with that, remiguring out, remembering what all of these things mean. LLMs are amazing at that because they understand the context around what this acronym is, and they've seen it a number of times. So the language, I think, is less of a problem. I think where the problem comes is in the specificity of the sort of experiment that the scientist is running. And I find this with a lot of creative tools with AI. One of the problems I often face is, say I want to ask a image generation AI to create some picture I have in my head. Wrapping that into a prompt is an impossible task because you have so much context that you're drawing on. You have all of these ideas in your head about exactly what it is you want the output to be. Once you've coaxed the AI and told it all of those things in order to get exactly the right output, you might as well have just done it yourself. You might as well have just created that thing yourself. And so I think the same problem applies here. And I think the thing that we're not saying very explicitly with what we're building right now is we can design your experiment for you. I just don't think we're not really that close to the AI being able to do that because there is just so much context. Maybe if you fed it in the results of the last experiment, all of the context that the scientists have, all of the objectives, I don't know, maybe you can get there, but I just don't think right now it's realistic. It's about how do you make the interface as seamless as possible so that the AI can do some of the grunt work and then you can take over and do the specifics. Those kinds of things I think are quite challenging with LLMs. But with the language, I think it's getting there, to be honest, and it's only going to get better.
These LLMs are so flexible in terms of the outputs that they can give you. And so I imagine that part of the work of incorporating this really flexible tool into your interface is trying to predict all of the approaches that the scientist might take when designing their experiments so that your system can handle all of the metadata that might come from those different approaches, but also being opinionated enough that you don't just have this sort of sprawling database of thousands and thousands of columns that have very little data in, trying to simplify the metadata so that you can capture the most important elements. So I'm curious if that challenge resonates with you, and if so, how do you navigate the being opinionated from a software perspective about the data structure versus giving the scientists the flexibility that they need within this very flexible tool, an LLM, to generate the metadata?
So when we started building this tool, we did a lot of research about, basically just read as many kind of experimental designs, as many protocols, as many methods as we could get our hands on, and tried to break it down into some kind of meaningful concepts that make sense both from the scientist's perspective, but also kind of are useful when you lay them out in a certain way that would then be useful from a sort of when you want to query that data later. And so that kind of formed the basis of the schema that we have for describing like how a protocol should be documented, like what are all of these different things. And so we have that as this like ground level, and it's very broad. It's not specific particularly to an area of science or even science in general. It's more of like a description of a process, but it's with science in mind and with some of the concepts of scientific experiments in mind. So that's the level of like opinion, like that's where we've been opinionated, is in that schema. Where we're not being opinionated is in what actually goes into that schema. So when the scientist decides, okay, I'm going to document my experiment and I want to write, so for example, I want to spin my cells down. We're not going to say you have to write centrifuge. We're not going to force a certain language or a certain way of describing an experiment onto the scientists while they're designing the experiment. What our kind of hypothesis is, and then the idea going forward is to say, okay, on the back end, that's something that we will deal with, because we're now in a place where we have tools which can automate the sort of standardization and like understanding of language that we didn't have 3 years ago. We can start to group and cluster all of these different concepts together and start to standardize them on the back end, so that when you want to query a database of all of this information, you can do so in a meaningful way. You don't have to deal with all of that flexibility. It's dealt with for you in the data structure and the way that we cluster things. But yeah, like the language itself is something we want to leave flexible, because I think otherwise the tool just becomes overbearing. Like it restricts the ways that scientists will express a lot of these things. And I think that can become really problematic.
What you just said is super interesting to me. Rather than forcing the scientist to apply a tag or to use the term centrifuge when talking about spinning the cells, you're planning to do post-processing steps that apply that tag on the back end, so that when you then want to query your data about all of the experiments that have centrifuging in them, that's something that you can do within the existing data model, without having to force the scientist to do that up front. Is that right?
Yeah, that's exactly it. Yeah.
Interesting. I know that you're a startup, so there's a plan at some point I assume, to sell the software system into biotech R&D organizations. How do you think about the challenges of bringing a system like this to market, especially when you're so focused on the needs of the individual scientist?
Yeah, it's obviously a question that we talk about a lot. Having this like inconsistency and incompleteness in how all the experiments are documented and all the problems that has for data teams. All of those problems, I think, are pretty like they resonate with a lot of companies. And so the key for us is focusing on like, how do we make sure that the tool is really useful for scientists, that they're going to use it, that the information that gets into the system is actually much better than it was before. If we can demonstrate that, and if scientists like using it and it's useful for them, then I think at that point we'd say, okay, great, let's see if we can start to implement this for an organization as a whole. But for right now, I think it's about making sure that we're really nailing the needs of the scientists and making sure that the tool is genuinely useful for them as an individual, and then building all of that collaboration, building all of that data. And I think selling into those companies is at that point should be much easier. That's our kind of approach right now.
If there's a scientist interested in working with the tool, or a company interested in prototyping the tool internally, what's the best way for people to learn more about you and the work you're doing at Briefly?
So our website is briefly.bio. You can find us also on LinkedIn. I have a Substack at Harry Rickerby where I've been writing about some of these things as well. If you want to follow, you can also sign up. If you're a scientist, you can sign up to start prototyping the tool. We're in like early access now. If you go to briefly.bio/builder, you can join the waitlist and we'll get you into the tool so you can start playing with it and seeing if it's useful for you.
Awesome. And just as we come to a close, are there any pieces of content, things that you've read or listened to recently that you think would be valuable for people in your arena to encounter if they want to learn more about this type of work?
The blog that I've followed for a while and has been talking about this space for a really long time is Jesse Johnson's blog, Scaling Biotech. He approaches it from the kind of working inside a biotech company, building a data team, figuring out the answers to all of these questions and how to build your systems and your team structure and your culture to tackle all of this. I think it's a really great resource. I would recommend checking that out if you're interested in this kind of stuff.
Yeah. So the podcast episode with Jesse should be out by the time this episode airs. For anyone who's interested in learning more about Jesse's perspectives, just scroll down in the feed and you'll definitely get to hear Jesse's thoughts on this arena. Harry, this has been a super fascinating conversation. I really enjoyed hearing what you're doing at Briefly and excited to see what's in store for you all in the months and years to come. So thanks for joining us.
Thank you so much, Ross. Yeah, it was great fun. Thanks a lot.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.